Yeah, so I think it's safe to say that proteins that can be designed to target discrete user-chosen DNA sequences seem to provide a natural fit for personalized medicine because they can really have profound effects on cellular phenotypes, therapeutically relevant effects, by binding to molecularly verifiable sequences. Today I'll talk about designed Zinc-Finger proteins. You may have heard of other types of such proteins like the CRISPR-Cas system, but I'll focus on Zinc-Finger proteins, which I think offer the real opportunity for high precision gene editing and regulation. And I use that term deliberately and in two different meanings, both in terms of being able to land the event you want at exactly the sequence you need to land it at in order to have the best possible effect, and also from the standpoint of specificity, being able to land on that sequence and nowhere else in the cell. So I'll provide a few examples. So the outline of my talk is listed here. I'll start with a brief overview of designed Zinc-Finger proteins, in particular Zinc-Finger nucleases and Zinc-Finger transcription factors, a brief description of what they are and how we design them. I'll then proceed into some programs where we are working to edit the genome therapeutically with Zinc-Finger nucleases. And finally, I'll talk briefly about our Huntington program, where we're using a Zinc-Finger protein transcription factor for addressing Huntington's disease. So this is a sketch of a Zinc-Finger nuclease. It's a programmable nuclease, again, very much like Talon's or CRISPR-Cas. This one contains two domains, the nuclease, the non-specific cleaving nuclease domain of the Fok-1 restriction enzyme, linked to a designed Zinc-Finger protein, which binds DNA sequence specifically, and that's the part of the protein that has the targeting capabilities. This protein works as a dimer, and so a dimer is shown here, and as such, it specifies an extended target. You have 15 to 18 base pairs bound on each side. You end up with a 30 to 36 base pair target sequence, and that's something that in general, relative to random genome sequence, you'll have a large number of changes, say, to the most similar potential random genome sequence. So it gives you the opportunity for a high degree of specificity. Below this is shown a Zinc-Finger protein transcription factor, or ZFP-TF, very similar to ZFNs, except it's not a dimer. It's a programmable transcription factor that contains two domains, again, a designed Zinc-Finger protein, and for the transcription factor, either a P65 activation domain or a CRAB, a repressor domain. Just a little background on Zinc-Finger protein DNA recognition, just to give you a sense of how we engineer these things. I'm showing you an expanded view of a single finger, or it's a sketch of a single zinc finger interacting with DNA. And the really critical thing to point out is that individual fingers, there's a 1 to 3 concordance between the binding element, the zinc finger, and the targeted region of DNA. And that's different, say, from Talon's or CRISPR-Cas, where it's more of a 1-to-1. One Talon repeat binds to one base pair. In a CRISPR-Cas system, one of your base pairs on your guide RNA is going to bind to one base pair. For us, it's 1 to 3. And the fingers specify their targeted region through a critical heptapeptide on their surface, which is in this amino terminal half of this alpha helix, such that we often speak in terms of the design of a finger and its binding preference, either a triplet or in some cases a quartet. Now you can make Zinc-Finger proteins by linking together multiple individual fingers that themselves each have been pre-characterized for binding to a 3 or 4 base pair sequence and making a longer protein with a predictable composite target sequence. And that's been used in the field with a good degree of success to make proteins. But at Sangamo, we use a slightly more complex version of this where we mix and match 2 finger units. And the reason we do that is that really it's this first bullet here. It enables more specific binding. Although zinc fingers are highly modular, they're not completely so. And there's a certain amount of crosstalk that you can have between adjacent fingers and their adjacent target sites. And this crosstalk can be beneficial if you pre-optimize these interactions to allow more binding cooperativity. They could give you more specificity. They could also be detrimental in a case where you put together 2 fingers that don't play well together and that either collide or try to reach into each other's target sites and specify different bases from their neighbor. And so if you make a 6-finger protein using this method to bind to an 18 base pair sequence, you can see how this approach yields 3 pre-optimized junctions and only 2 novel ones, as opposed to if you were linking 6 individual fingers together and you'd have 5 novel junctions, greater jeopardy for a bad interaction and harder to optimize your specificity. So this is what our design process looks like. We will go through a target sequence looking for 18 MERs or 15 MERs that we can recognize. and then link together corresponding 2-finger units or a small panel of 1-finger units if we're in the case of a 5-finger protein. Link them together and get something that binds to the corresponding composite sequence. Now, the complexity in this approach is that if you're using a 2-finger unit, you have a much larger panel of subunits that you have to make. Instead of 64 triplet binders, you have to worry about thousands of hexamer binders. And the company has spent a lot of time in its history working on developing such an archive of 2-finger units. And at this point, we have thousands of 2-finger units for hexamer recognition derived from many different sources, and they're highly specific for diverse sequence compositions. This is just a set of 4 such 2-finger units and some summary biochemical data. This is a summary of a base frequency matrix from a CLEX assay showing that we can get very good base preference across the 6 bases of their target sites. Okay, so that's sort of the main principle for zinc-finger protein design, working on these modules and getting modules for as large a fraction of this hexamer space to bind as well as possible. But an important extra factor in design is highlighted in the following slides, and that is you also can gain a lot of power by diversifying how you can link together these 2-finger units. So if you take the top part of the slide as the standard zinc-finger nuclease configuration, this is as developed back in the 90s. The key thing to point out is that the fingers are adjacent to each other. There's no gap between these colored hexamer sites. And the binding sites for the 2 proteins are separated by 5 or 6 base pairs. That's the standard parameters as the platform was developed back even before Sangamo was founded. What we've done in order to increase our design capabilities is introduce the possibility of base skipping linkers between the individual modules, which, as you can see, allow you a much greater flexibility in terms of what hexamers you have to recognize to land a Fok dimer domain in a given location. Right? If you really want to land your dimer and cleave here, if you don't skip bases, you have one chance. You have to bind well to this brown, yellow, and orange hexamer, and if you can't get one of those, then you're not going to get an optimally binding protein. But if you can allow yourself to skip bases, you now have more shots on goal, more hexamers you can work on. And I should mention that these base skipping linkers, these are highly engineered, rigid linkers with actually with very high proline content. They were developed by phage display selections. They're not sort of flexible glycine serine tethers. They specify a gap, they impose a skipping, and they have very little entropic binding cost, so they work quite well. So you can see how if you allow yourself to skip bases, you get many more configurations per dimer for landing a Fok domain dimer in a given location. Likewise, if you developed alternative linkers for linking the zinc-finger protein portion to the Fok domain, you can allow a wider range of gap sizes. Instead of 5 to 6 base pairs in the parental architecture, you can now have 5, 6, 7, 8, or 9, and this allows you more opportunities to land this cleavage event exactly where you need it to be. And also, if you reverse the Fok-ZFP polarity, you can also improve things. This is the standard Fok attachment point on the carboxy terminus. If you move the Fok domain to the immunoterminal side of the ZFN, you go from having just one dimer that you can make for a given location with the right-hand target on the top strand, the left-hand target on the bottom strand, to 3 more where both targets are on the top strand, the bottom strand, or top and bottom strands but reversed. And I should point out, again, this is a case where the field originally started with the linker over here. This is a much easier attachment task. If you do a 3-dimensional docking exercise, you find that the relevant endpoints of the 2 domains are close to each other here, and it was a fairly straightforward development for the field back then. Immunoterminal attachment points are much more difficult. There's a longer distance and some strange twists that the linker must take, but we were able to develop that using a bacterial selection system. Okay, so what does this get us? So between the large panel of 2-finger units that we have and the a diversity of ways that we can link them, we now have a very high targeting density, a very high targeting precision, as you were, an ability to land a cleavage event pretty much at any location of our choosing. This is an example, a globin locus test sequence. It's a 50 kb sequence from I believe the beta-like globin genes. And we use this to test our design algorithms. This represents 50,000 potential sites of cleavage, right? So as you ratchet a cleavage domain 1 base at a time through this region, you can say that there are 50,000 discrete locations where you might introduce a cleavage event. And we could target 30,000 of these, or about 60%. And important to point out that this actually compares very nicely with, say, some of the high-fidelity Cas9 systems, where you're limited to a much lower fraction, down around 2 to 5%, because they have a much larger number of base restrictions in what they can target. Okay, so that's a brief introduction to the technology. I'm now going to provide a couple of examples, the first of which will involve therapeutic genome editing with zinc-finger nucleases. And before launching to that, I just wanted to provide a brief overview of the two types of gene editing pathways that ZFNs, or really any targeted nuclease platform, can enable. Once you get a DNA cleavage event, the cell can take one of two fundamentally different repair pathways. If there's a repair donor around, this can be something that the experimenter adds, or it could be a homologous chromosome or a sister chromatid. You can get repair using homology-directed repair. And this can enable the correction or addition of genetic material by virtue of repair using these homologous regions. If the cell doesn't use this approach, it will do another pathway called non-homologous end joining, which is essentially the cell just taking these ends and gluing them back together, which is frequently error-free, but will often enough yield a few bases of insertions or deletions such that you can use this approach to get a functional knockout, especially as the cell, if it does repair it error-free, you can cleave it again until the cell finally does create an error, and then you stop cleaving. So with this high precision targeting capabilities, we can get homology-directed repair to proceed very well. I should mention, I forgot to mention, for this left-hand pathway for HDR, it's really important that whatever base, whatever event you're trying to correct, actually it's probably better to show it here, whatever base you're trying to repair or genetic change you're trying to make, it occurs in a much higher efficiency if the intended site of change is as close as possible to the center point of your cleavage. So if you want to, say, make this change from a G to an A in order to do the study of early onset Parkinson's point mutations, you're going to get it most efficiently if your dimeric nuclease straddles this site and is centered on it. And as you can see, we're able to develop ZFNs that do just that. Likewise, for collaborations for alpha-1 antitrypsin targeting the PIZZ mutation, as well as collaborations targeting the sickle mutation in globin, we're able to get cleavage events that occur at or almost, if not at, but adjacent to the base of interest, allowing very high efficiency HDR. So there are moving to the other pathway, non-homologous end joining, there are circumstances where you want high precision for knockout applications, where you're not trying to do the homology-directed repair, but you are instead trying to introduce some small insertions and deletions or indels into the targeted region. And the example I'll show is from the LCA10 mutation in the CEP290 gene. This is an interesting gene, an interesting condition in the sense that the disease mutation is an A to G substitution here within an intron of the CEP290 gene. So it's not what you would think of as your sort of standard point mutation type disease where you're changing the coding sequence. Here you're in the intron. And this mutation creates an aberrant splice site. So there's a 5 base segment in here that's almost a good splice acceptor, but in a wild type sequence, the transcriptional apparatus will just pass it by and not use it to any significant degree. But once you make this single base change, it now becomes a much better splice acceptor, and that causes problems because you get this exogenous extra exon spliced in, which is non-functional and messes up the gene function. And what this does is it leads to progressive vision loss. This is important for vision. So the goal here is to make a mutation here that really anything will do. Knocking out a few bases will disrupt this novel this novel splicing site, and will disrupt its function, allowing proper transcription, non-insertion of this aberrant exon and proper function. I should mention also that this was an interesting gene for us to target in the sense that this was a gene target for Editas, which is one of the CRISPR-Cas companies, and they actually had trouble cleaving right here. They have to go with an approach that cleaves twice, once to the right and once to the left. So in this case, we were able to develop ZFNs that target this base fairly precisely with a cleavage center point centered on the mutated base. We're able to show that we get good indel production from treatment with these ZFNs. And we can get 85% indels really in a first pass design effort. So in 2 weeks, we can go from a design set all the way through to reagents isolated that modify this quite well. So you can see how this possibility for speed could allow you to rapidly develop reagents for novel mutations or novel sequences. As a second example, I'll talk about targeting BCL11A for treatment of beta hemoglobinopathies. BCL11A is a transcription factor found in hematopoietic stem progenitor cells and the associated cellular compartment. It represses gamma globin in the erythroid lineage, so that's just highlighted here. And erythroid expression is controlled by a key GATA site. So what happens is during erythroid development, during lineage development, what will happen is BCL11A is upregulated, and that will then in turn cause the downregulation of gamma globin. And importantly, this protein is responsible for gamma globin suppression in adults. So this is a sketch of the various globins that you make during development. You originally start off making epsilon development at a very young prenatal age. Then during development, you start making gamma globin, which is high until just after birth, after which that's downregulated and beta globin comes on. And what this means is that people with beta globinopathies, like sickle cell disease or beta thalassemia, they're okay when they're born. It's when the switchover occurs and their good gamma gene is suppressed and their bad beta comes on that they have problems. And so that raises the possibility of addressing such diseases by setting up a situation where the gamma is kept on, and so the good gene is retained. And in fact, there are naturally occurring human variations where people have persistence of gamma globin, and if these people happen to have, at the same time, a bad beta globin gene, they have a much better course of the disease than if their gamma is turned completely off. So the goal here is to knock out the erythroid-specific enhancer of BCL11A. I should mention that BCL11A has other essential functions in other compartments of this differentiation scheme mentioned here or shown here. So you can't knock out the coding sequence. What you want to do is knock out that control element, which is specific for BCL11A upregulation in this lineage. And so we targeted this GATA factor binding site. We were able to get ZFNs, again, that straddled the element with the cleavage point centered within it. We're able to show that if we take CD34 cells and we treat them with the ZFNs and then run the cells through an in vitro erythropoiesis process and measure gamma globin, we can show good upregulation of gamma globin going from a GFP-treated cells to this orange bar here where we're targeting the GATA site. And just as a comparison, we're almost as high in upregulation if we did a BCL11A coding knockout, which again is not a valid approach for therapeutic purposes, but a nice positive control. I should mention we published this a couple of years ago in Nature Methods. You can take these modified cells, and you can, I should point this out here, you can take these modified CD34 cells, edit the BCL11A enhancer, and then engraft into immunodeficient mice, run these mice for 16 weeks, harvest the bone marrow, and then perform your in vitro erythropoiesis assay and show that you also get upregulation of gamma globin as determined via TECMAN or HPLC of the proteins in the resulting red blood cells. So for these approaches, for really any gene editing approach or gene regulation approach, it's important to gauge the specificity of the proteins that you're making. Of course, it's always good to have high on-target function and desired biological effects, but there's always the question of, well, what else are you doing in the genome? And for that, we and really the rest of the field turn to this assay, commonly known as the N-capture assay. Some of you may have heard of this as GuideSeq is one incarnation that's in common use in the field. And this is an assay we're spending a little bit of time on this slide because it's a really important one in the field. So this assay is developed to answer the question of, again, you've treated cells with your nuclease. You know you have your on-target working because you know to look there to see that there are mutations occurring there. But where else has it cleaved? There might have been these fleeting cleavage events elsewhere in the genome that were around for a little bit of time and that have been repaired, possibly misrepaired. How do you find that? And so the way that we go about this is you take a cell line, in our case K562 cells, that tolerate having a high amount of oligonucleotide duplex around. And you start off by treating those cells with your nuclease. And what will happen is, under those conditions, a fraction of those cells will get integration of this donor oligonucleotide duplex into the breakage site. It doesn't happen to all the sites, but maybe about 15% or so, a high enough amount that you get pretty consistent tagging of anywhere that's going to be cleaved. Once you have this integration event, this now allows you to lay down a primer on that, that known sequence. And through some fairly intensive molecular biology, you can amplify up this genomic sequence that flanked the integration event. You can sequence those sequences, and now you have candidate sites for where your cleavage occurred. And I mentioned that they're candidate sites because while this assay is very good at finding real off-target sites, it does have a false positive rate. The molecular biology involved in this step is fairly intensive. It can yield candidate off-target sites that are not truly cleaved. And so it's really important to then do a follow-up study in which you do a second step where you take the candidate sites that have come out of this part of the assay and you go into cells that are therapeutically therapeutically relevant or relevant for whatever application you're doing, and you test for those cells to see whether you actually see cleavage at the candidate target sites. And in this case, we're developing this as an ex vivo therapy. We're going to be treating a large number of CD34 cells with RNA delivery by electroporation, and so we did our follow-up studies in exactly that system. So what do we see? So when we do the capture studies, all the capture studies generate results that look broadly like this. We get a set of loci, and I've just given you locus rank number, the top one of which is the on-target site. And we get an integrant count, a number of sequence reads that came from each of those loci. And so what you ideally want to see, of course, is your targeted locus at the top of the list, and then a steep drop off and hopefully background levels further down. And I can tell you from prior experience with this assay, this is a steep drop. And these levels are consistent with background levels that may or may not be real sites, but are certainly consistent with them not being real. When we do the follow-up studies, where we look at indels in ZFN and control-treated CD34 cells, we find that we can modify these cells at a very high level, 82% indels. And even at this very high level of on-target modification, when we turn around and look at the off-target sites, we see no loci modified significantly, suggesting a very highly specific reagent. One important thing to point out is that in these specificity studies, we use very high nuclease levels, both for the capture protocol and the follow-up analyses, very high percent modification. I point that out because there's a lot of specificity is an important consideration for developing of any type of therapeutic nuclease. And there are a lot of presentations and publications that will point to no measured off target. But it's really important in viewing these presentations and reading these publications to ask, well, okay, but how hard are you pushing your system? It's a fundamentally different result to say you found no off targets and you modified your on target to 10% versus say modifying your on target to 90%. And so I'd like to really emphasize that these reagents were pushed quite hard. Okay, so as a second example, I'm going to talk briefly about the T cell receptor alpha constant region, which we targeted. This is a target of interest for immuno-oncology. Essentially, if you are taking T cells and modifying them with a CAR or with a novel T cell receptor to target cancer, it can be beneficial to knock out the native T cell receptors that these proteins express. So in this case, the goal was to target the exonic region of this protein, this constant region. It's 3 exons, 374 base pairs, 374 potential sites for cleavage. We can target 218 of those using ZFNs, or about 58%. And this over here, this gives you a sense of how our target sites spread throughout the 3 exons. The exons are shown at top here, and you can see that there's clouds of targets which really don't yield any gaps, and that's what I'm trying to show here. If we take a subset of these candidate designs and we test them for modification of their target sequence, we find that in each of the exons 1, 2, and 3, we can find highly active pairs. These are, in this case, we're testing them in K562 cells delivered by plasmid transfection. We're able to see 55 to close to 70% modification, so very, very good for an initial scan. We can then take these proteins and test them for how well they knock out the T cell receptor alpha constant region in T cells themselves. And for that, we use a FACS assay. And for this, we deliver the ZFNs using RNA delivery. And after 10 days, we assess for T cell receptor alpha disruption via sorting for CD3 surface expression. And what we can see is that in non-treated T cells, we get the expected FACS plot with with really essentially all cells positive for CD3 surface expression. Once they've been treated with ZFNs, we now can get up to 90% plus of these cells that are negative for surface expression of the T cells. Okay. I should point out that these ZFNs are highly active and well tolerated. Let me just gather my thought for a second. So really important to point out here is we can get these very high degrees of modification, excuse me, in spite of the fact that these cells are grown for 10 days post-treatment. Very important to point out because you can often get nucleases to highly modify target genes, but the really critical thing is, are you beating up the cells while you're doing this? And if so, what you'll see is a decay of these signals over time. And so we're able to see a very high degree of modification retained 10 days out. So in the interest of time, I'm going to skip through these next slides, except to say that in order to confirm specificity, these reagents were also tested by the assay I just went through with our BCL11A program. And just to point out that we again obtained very high degrees of on-target modification with no significant off-target modification. Okay, so I'm going to skip to the last part, which is to describe zinc-finger protein transcription factor approaches for addressing brain disease. So as I pointed out at the start of the presentation, the platform can not only provide for zinc-finger nucleases, but also ZFP-TFs, or zinc-finger protein transcription factors. And we have developed zinc-finger protein transcription factors for targeting the CAG repeat tract of the Huntington disease gene. So just as a reminder to everybody, the Huntington disease is caused by an expansion of the CAG repeat tract such that in unaffected wild-type people, you have a CAG length that's in the teens or in the 20s, and that's what the naturally occurring gene has. In patients, you have an expansion of this tract to upwards of 30, 40, sometimes even larger numbers of CAG repeats. And this causes in a dominant fashion, it causes neurodegeneration. And so we were interested in developing ZFPs that could bind to this CAG tract in a way that was cooperative, such that you got very highly efficient binding to this enlarged tract, but very little binding and very little function at this the normal size tract. We set up a system where we can monitor for this by looking at expression by taking advantage of downstream SNPs. So we make TACMan reagents that would be specific for one SNP or the other in either the disease allele or the wild type allele, and that's how we were able to differentially monitor the two alleles in a patient cell. And what we showed is that we were able to make ZFPs that could do exactly what we hoped they would. It's really best to just focus on the dark blue bar and the red bar corresponding to the wild type and the expanded allele. With allele-specific repressors, these are proteins that bind to a succession of these CAG repeats, a 5 or a 6-finger protein. We were able to show good, near 100%, probably about 95% repression of the expanded allele with full retention of expression of the normal length allele. And just as controls, we showed that we can throw in biallelic repressors that bind to other parts of the gene that repress both alleles. Also, I should point out what the light blue bar is. The light blue bar is a way of detecting both alleles at the same time, and we see a nice drop by a factor of 2 in that signal. We also followed this up, probably down to about 2 minutes. We followed this up with protein studies where we looked at the same information, but looking at protein expression, monitoring either the expanded protein or the normal size protein, and we could see a differential repression of the expanded allele protein. Now, patients have a wide range of splits in terms of their wild type CAG repeat length and their long CAG repeat length. Most patients are in the 40s with their long CAG repeat length, and we showed that over a variety of splits of different short lengths and long lengths, we were able to get differential expression over wide titration ranges and a wide range of patient fibroblast cell lines. We took our reagents for the CAG 6715, we took the 6715 cell lines and tested proteins by Affymetric studies to see how specific they were. And what you're really looking for in this study, this is called the volcano plot, where we're plotting fold repression on the x-axis and p-value on the y, and you're looking for an absence of things that are here that are more than twofold repressed and that are not significant in this area that is twofold repressed and has a significant p-value. And what you see is this area is largely free except a couple of hits right on the edge here. I should point out that Huntington does not show up in this assay because the Affymetrics probe that's used for Huntington does not differentiate between the wild type and the disease length repeat. So that would be down in the, probably in the noise here, just below twofold repressed. Okay, so just for a final set of slides, just to show how these proteins behaved in more sophisticated systems. We wanted to take them into neurons and then in vivo. So here we're working with embryonic stem cells from patients and differentiating them through neuronal stem cells, finally to neurons, and looking at how we could repress the disease allele differentially with mRNA delivery and assaying at 48 hours. And as you can see, in all these systems, we could get very good differential repression of the disease versus the wild type allele. Once we had these differentiated neurons, we could treat them with an AAV vector, since these are not dividing anymore, and show that we could get repression staying stable out to 21 days. And in fact, with lentivirus delivery, really this should connect back to the stem cells because we did the infection at this stage of the process. We're able to run these cells now for 103 days where they're stably expressing our repressor proteins and differentially repressing the expanded allele versus the wild type allele. So proteins that are very effective for their purpose and very well tolerated in the sense that, again, if you have a stable expression of a protein that's impairing cell function, what will happen is over time, its non-transfected brethren cells will overgrow it and the signal will go away. So very well tolerated proteins. Okay, so for my last two slides, we evaluated these zinc-finger protein transcription factors in mouse models of Huntington disease, either this HDHQ50 line, which has as one of its alleles the human exon 1 50 repeat gene, and this R62 line, which has a much larger repeat. It turns out that in mice, the repeat lengths characteristic of humans don't cause disease. You have to go to a much larger repeat length to see some disease phenotype. And so we work in both systems, one to look at molecularly, are we getting the recognition that we want in binding to the disease, the more typical disease length repeat tract, and in this one to see whether we're having useful phenotypic outcomes. So in this case, we were able to show with the first model system, we were able to show 50 to 80% of the repression of the Q50 knock-in allele throughout the mouse striatum, and that's just shown here. So this is striatum-wide. There's no selection for modified cells or nothing funny going on with the analysis. This is just taking whole striatum, grinding it up, and showing repression 7 weeks after treatment. On the other hand, with the Q7 allele, we show no repression. And finally, in the R62 mice, which do suffer from phenotypic changes, we're able to show that we're able to differentially repress the disease allele, as shown here when in ZFP-treated mice. And we're also able to get upregulation of biomarkers of medium spiny neuron health and just presence. So the STARP32, PDE10A, DRD1A, DRD2, we're able to show that relative to GFP-treated mice, we're able to show upregulation in the ZFP-treated mice. And finally, there are behavioral studies that you can do, clasping phenotype and other types of assays, open field tests, where we're able to show that treated mice develop these disease-related phenotypes with a lag and to a lesser degree than their untreated controls. Okay, so just some take-home points. So zinc-finger proteins provide, I think, a robust and versatile platform for enabling sequence-specific therapeutic interventions. Targeting density of the platform is quite high, so you can really land your event, whether it's a regulation event or a cleavage event, essentially wherever you want. Optimized ZFNs yield highly efficient cleavage with little or no off-target effects, and likewise, ZFP-TFs can enable repression that is potent, specific, allele selective, and really very durable and well tolerated. And the platform has been validated as safe in the clinic We have additional trials underway, so this is a sort of a brief overview of some of the key trials that Sangamo is involved with. I spoke today about these 3 programs. We also have programs in hemophilia B, MPS 1 and 2, as well as a straight hemophilia gene therapy program. And I just want to acknowledge colleagues and collaborators who helped make this work possible. So thank you.